excited to see you all here. This is our first impromptu debate event. We're super proud to have you both in the room, Yuri and John. So we're gathered here to figure out how do we defeat death, right? It's an important question. I don't want to die. I know many of you don't want to die too. So how do we do it? How do we solve it? So we have two brilliant minds in the room, each of them making great progress in their field. Partial epigenetics, brain tissue replacement, great stuff. But what actually works, right? Like, let's find out which idea has the strongest hold and can get us closer to not dying. All right. So the format of this is we're gonna get 10 minutes to each of the speakers to pitch their idea. Right? So, impromptu 10 minutes, make a great point. After that, we're gonna open this up to questions to both of the speakers, to each other. 2 questions to each, right? So, provocative questions, 30 questions. 30 questions.
30 questions. Is this
right?
a scientific debate or
scientific debate, but we want to get to the core.
Right.
We want to find out what's going on. Then afterwards, we will open questions to the group, to the public. And how are we going to decide on this? So we have 3 judges in a room that will vote 2 times and give 2 votes. So the first vote will be for the presentation. So the judges will vote for each of the pitches they believe the most. And the second vote will go towards the debate. So the judges will vote on who, in their opinion, won the debate, right? With the best to answer questions. So that's how we're going to roll. So in order to decide who rolls first, we want to toss a coin. So head or tails?
Heads.
Heads? Tails? All right. Let's do it. Okay. This is heads.
Okay.
All right.
I'm going first.
Heads.
Okay.
You're up. Okay, let me. Hi.
All right.
I'll set up a timer. So we're
Okay.
checking the time.
So I guess, just to set the right format, I guess it's less about what's working right now and more about our maybe differing views on aging and how aging works, because obviously right now we don't have therapies yet that work for humans, and it's about the approach that each of us is picking. Replacement approach or partial reprogramming, epigenetic degeneration or partial reprogramming approach. And I guess the idea for the debate came up with just to discuss from each of our perspectives how we think aging works, so that we're on the right path to defeating aging, because maybe one of these perspectives is incorrect and so could lead us to a dead end. Now that said, personally I'm all for different approaches, trying different things, because obviously we can debate things, but we can be wrong. Both of us can be wrong. Aging might be something completely different, and it's great to have different people attacking aging from different angles, so that eventually we have potentially at least something working out. Now, in terms of, I guess the perspective of replacement versus reprogramming comes from the idea, at least from my understanding, is whether aging is programmed or whether aging is totally random. And at least the way I understand John's perspective is that he believes that aging is totally random and driven by stochastic changes or non-enzymatic changes to long-lived molecules, such as like the molecules that comprise the extracellular matrix. And then that matrix produces some sort of signals that tell the cells within that matrix, within the organelle, within the organism, that they're old. And one of the examples that Jean used is that if you take young cells, put them in the old matrix, and that makes those cells behave like old cells. And conversely, if you have old cells, but you put them in the young matrix, then those cells start behaving as young cells.
And to me, actually, this is already an interesting observation that basically means that cells can interpret external signals and behave differently, and so we actually can potentially override the signals. So even if we're unable to change the matrix, we can maybe change the cells by partial reprogramming or some other methodology to actually ignore external cues and fool them into they're young, or make them behave as if they're young, upregulate their gene expression of the good stuff, downregulate the bad stuff, basically shift the pattern of their gene expression towards their younger pattern. And this is really the idea behind partial reprogramming, where we take human factors and try to convince the cell to shift its gene expression pattern, try to influence the cell to shift its gene expression pattern towards that observed in younger cells. But I think more philosophically, from my perspective, I think aging looks more as if it's actually programmed than completely random, because as I showed in my talk yesterday, if we look at the animal kingdom, there's so much variation in lifespans and the speeds of aging that, to me, I think that all this variation, which is caused by randomness, and including the observations that epigenetics can influence even within a single species, greatly influence lifespan, and for example, according to some external cues, can greatly expand lifespan or shorten lifespan. Like my favorite example is the monarch butterfly, that if they're born in the summer, they only live 1 month, but if they're born in the fall and they need to go to Mexico for the winter, they live for 9 months. So obviously there's great modulation of lifespan, and nature has this ability to have much longer lifespan, but sometimes it chooses to shorten the lifespan of its species. And also another observation that to me points to at least the signaling, if not programmed nature of aging, is the observations that we see with parabiosis, where we connect a young individual and an old individual together in their blood circulatory system, and we see that the old individual is rejuvenated while the young individual is actually aged. But then you disconnect them and they return to baseline. And to me, this shows that aging is reversible, at least the aging in that model system. So it's not necessarily just stochastic accumulation of damage, because if you rejuvenated the old organism and you separated it, but then it returns to baseline, then obviously somehow we were able to reverse that damage that potentially, we think should be reversible. Because we think about aging as just stochastic accumulation of non-enzymatic damage to long-lived molecules. And another observation, again, to me that talks against this hypothesis is that if you transplant organs, even young organs, to an old organism, you see very quick aging of these organs. And so it's not necessarily the matrix that ages them. Because if you take like a whole heart transplanted into an animal, and you see that heart being aged, obviously the heart came with its whole matrix. But somehow, if you put it in the whole organism, it very quickly synchronizes with the age of the organism. And so to me, this implies that there's some sort of signaling, systemic signaling that ages that organ, and not necessarily just random damage that is at work there. And so basically, all of that preamble means that for me, aging is driven by epigenetics. And so if we intervene at the level of epigenetics, not only would we be able to reverse this kind of signaling that tells cells you're old, but even if in the context of the matrix, we could also potentially tell cells to ignore the matrix signaling. So essentially we're getting like the best of both worlds by intervening at the level of gene expression. Then ultimately, of course, cells, they do the work, and they do the work at the level of proteins that are expressed in those cells. And so if we are able to force the cells to ignore external cues, we can potentially rejuvenate it by the approach of some sort of epigenetic modulation, of which partial reprogramming is one such approach. But of course, that said, I welcome trying other approaches as well, including the replacement approach. Oh, but one thing I'm worried about the replacement approach is that if aging is driven by systemic signaling from the brain, and the brain has the control center that actually synchronizes the rest of the tissues, then replacement would not work, because if we transplant the head on a new body, that old head might actually quite quickly age the rest of the body. And so a key proof of concept experiment in this case would be switching heads between an old individual and a young individual, mice, of course, for now. Essentially, it's like heterochronic parabiosis, but actually heterochronic head transplants, and to see would the old head rapidly age the new organism. If that happens, then replacement might not help us. Or, and conversely, would a young head rejuvenate the old organism? And this will be conclusively then telling us that there is this systemic signaling happening in the brain that can either rejuvenate the body or quickly age the body. And this will serve as a convincing experiment showing whether the replacement paradigm is viable or not. So I guess, I don't know how much time I got left, but I guess I can yield the floor to John.
Okay.
Okay, I'd like to thank Micah for arranging this and Yuri for your willingness to discuss and debate these points. It's really not about us. It's about learning what we know about the science and what we don't know about the science. And I think that's super important. And we need to get down to the bottom of a lot of what you said, because I think there are a lot of assumptions there, not the least of which is calling a change in epigenome rejuvenation. I think that's a big problem with the reprogramming field. Rejuvenation without maximum life extension is really meaningless. And so the evidence that partial reprogramming extends maximal lifespan is essentially non-existent for normal individuals. Normal individual mice. Right. So, there's the progeria example. So I'll start maybe just with replacement, and you can come back, please, with some of the points you raised about replacement. But I'll work on the assumption that if we replace all the parts of our bodies, the old parts of our bodies with young parts of our bodies, we will get rid of all that time-dependent damage that is accumulated in extracellular matrix, in DNA, and other components of the body as well that haven't been looked at yet, like lipids and carbohydrates, for example. So if you rebuild new tissue from scratch, it won't have any of this time-dependent damage. So let's assume, and again, if you have issues with that, you can come back with that, but let's assume that replacement will get rid of all this damage. So if you were, for example, to push it to the extreme, take an old body, replace it with a young body, you would have a young body. I don't think there's any evidence that the brain controls the rate of aging of the body. But anyway, you would also replace the brain, not just the body. So everything would be replaced. I don't. If you have an argument that wouldn't work, then let's debate that. But let's assume that works, and then I'll spend the rest of my time sort of on partial reprogramming and what are my issues with that. So partial reprogramming, for one, I think doesn't deal with a lot of what we know ages us, and that is like, for example, the extracellular matrix. Some would also say DNA damage is a primary driver of aging, and epigenetic reprogramming doesn't influence either of them, as you've readily admitted, I guess, that it doesn't address the type of damage that accumulates in ECM. Your assumption there is that we could ignore the ECM. Cells don't need the ECM to live happy, productive, functional lives. I mean, that goes against everything we know about biology. Right? The ECM of cells is intimately related to their function at all times in terms of their ability to acquire nutrients from the circulating blood. Metabolism in general is highly dependent on ECM. So it's not, I make no assumption about how the ECM deterioration affects the health of the cells. We just know that it does. So the cell, you can make the cells ignore that, but that's the symptom. That's not, you're basically ignoring aging at that point. Those cells will, for example, blood vessels, right? The stiffening of vessels in the body, we agree that happens with age. That's in the blood vessel walls, that's ECM. That damage alone will kill us. We can't just ignore it. Rupturing blood vessels is not going to allow us to live very long, or ischemic or hemorrhagic strokes are not going to allow us to live very long. So if partial reprogramming doesn't address the ECM damage, we will die on schedule, with this type of damage. It also doesn't address DNA damage, so I don't think that's as big a driver. There's not as much evidence, in my view, that DNA damage is a central driver of, or limitation to maximal lifespan, but it probably contributes. But epigenetic reprogramming does nothing for DNA mutations. Right? I know there's one paper that looked at histone 2A phosphorylation, that's double-stranded breaks, but again, no sort of an indirect method, one type of DNA damage, and there's caveats with that study too. So really, there's no evidence and no reason to believe, more importantly, no reason to believe that partial reprogramming will have any impact on maximal lifespan, because it has no effect on what we consider aging. So if you look at a 20-year-old and a 90-year-old, you can see the difference. What is that difference? It's skin deep, but it's true for every part of the body. If you look at their brain, the ECM is completely degraded. If you look at their skin, you look at any organ, the ECM is completely degraded. And if we ignore that, you can fool the cells into thinking they're young for a little while and maybe boost performance in certain measurements, but you're still gonna, you can't have those cells continue to live in an environment where they don't get proper nutrition, oxygenation from circulation, because the blood vessels are falling apart, or everything else is falling apart. So it's I think we should debate some of the other points in more detail as well, but essentially I guess that's the sum of my perspective.
All right, I guess to go back to the
Yeah. So let's get one question from you, the answer.
Okay.
Question.
maybe address some of the points.
Yeah. If you want to address something, do we want to give the judges a chance to vote first? Okay.
Very experimental format. There with us.
Yeah.
Misha proposed to have more debates and philosophical topics yesterday, and the distance between proposition and action is
It's minimal.
very short
Yeah.
iterations.
Okay.
Vote after there's a bit more discussion.
You want to vote after?
Yeah, give people a chance to have their discussion.
Okay. Then, let's continue the discussion.
Окей. Тогда...
There's a debate about the debate.
Тогда давайте сейчас они зададут вопрос, а потом оцениваем.
All right. So let's continue the debate, and then after that, the judges will vote on the debate portion of this, and then there will be a question moment for everybody, and then we'll vote on that separately. Does that sound good? Okay. 2 questions from each other.
Okay.
Yeah. You want to start?
Oh, questions?
Or you want to address something?
Yeah.
Because there were questions asked yesterday, right? Or rather, more so in that.
Is it just some points about partial reprogramming?
Yeah, if you want to address those.
Right, in terms of maximum lifespan, I guess, there is one paper on maximal lifespan increase in normal mice from Rejuvenate Bio recently. They used an AAV approach, like a gene therapy approach. They injected really old mice. They saw an increase. I mean, it's not a huge increase,
No,
but
it's not.
yeah, but at least from a proof of concept standpoint. And of course, in progeric mice, there was a substantial increase in both median and maximal lifespan. But I fully agree that there's still a lot of work to be done to demonstrate that partial reprogramming can extend lifespan in normal mice. And I think right now we're just limited to partial reprogramming being in need of a tissue-specific approach. I mentioned yesterday this preprint from Alejandro Campo's lab showing that there's some organs that really limit our ability to induce reprogramming for too long, because if we reprogram for too long, they start failing, like the liver and the small intestine. So we need to figure out how do we avoid these organs that can cause problems and yet push other organs a little more to get the rejuvenation, and also figure out kind of the cadence, like how frequently do we use reprogramming, because there's some evidence that it's not just the reprogramming process that rejuvenates stuff, but actually it's the time afterwards, once you stop reprogramming and the cell kind of returns back to its original state, that also could be the moment of some measure of rejuvenation. And so we need to figure out, maybe we're doing it too frequently and we need to do it less frequently.
And by rejuvenation, you mean epigenetic?
Well, not necessarily just physiological rejuvenations. Yesterday I showed, there are a couple of papers showing that it's at the level, also at the level of transcriptomics. So the gene expression pattern looks like a younger gene expression pattern, but also the physiology of some tissues that they reported, like the spleen or the liver, or actually I think some skin also reported physiological improvements. But yeah, in terms of lifespan, definitely like the gold standard, if an intervention is successful, is lifespan. So.
Yeah. Can I respond to what you said?
Yeah,
so
sure.
far? Yeah, so the papers, right, it's like a very, very minor maximum extension. And we've seen much more, for example, with caloric restriction, and many more papers. But even that is very weak evidence, right, because it's strain dependent. It depends on the controls you're comparing it to not being ideally healthy. So these are, these are arguably not significant or meaningful improvements in lifespan. The progeria one is like a very specific mutation. It's nuclear laminin A that disrupts expression of genes. So yeah, maybe you can have a benefit by having transcription factors in there that somehow normalize gene expression. But that's a very specific disease that you're fixing, addressing correctly, sort of the problem that it's a one gene mutation, right? And it disrupts gene expression, and so it's not necessarily surprising. It's good. So epigenetic reprogramming, I think the evidence might be strong enough to pursue that as an interest to treat Hutchinson-Gilford progeria, but that doesn't mean that it's reversing or slowing aging. It's just treating a particular phenotype that happens to share features with aging. But of course it would, because lack of normal gene expression will lead to all sorts of deficits in function that mimic aging, because everything also in aging goes downhill. So I don't think that's any evidence that you're slowing or reversing aging.
Curious how caloric restriction, in your view, extends lifespan by preventing, I guess, damage to the matrix. Like how does caloric restriction?
No, I don't think it has any effect on it. I think it's just that the mice are healthier when they eat less, right? So they have less.
Starved, right? Actually, like
they're not starved, because starvation shortens lifespan.
every other day.
But they're fully nutritionally fed, everything they need with no excess.
So you don't think caloric restriction actually extends lifespan? You think it's just
It
normal diet
extends
shortens?
it extends median lifespan, and that's across species. It's pretty universal, but it's not. It doesn't extend maximal lifespan in a significant way.
In mice and rats, I'm pretty sure it does.
Well, again, you're comparing it to controls that, and not all strains of mice, only some strains of mice. Some strains, it shortens lifespan, so it's strain dependent. Depends on your genetic background. So, and that's the best that the longevity field has come up with, is caloric restriction so far. And it's really weak evidence that it actually slows the aging process. It extends medium lifespan, yes, because it makes them healthier, but not maximum lifespan. Or the evidence isn't very good.
So wouldn't
And
that
at best mixed.
speak against this being, I guess, preventing damage from occurring, rather than maybe some sort of like inherent program that extends lifespan for some strains when we detect
Yes,
like adverse conditions?
the problem is no one looks at the damage except, biochemists when they first were studying aging looked at the damage that occurs to these long-lived proteins over time. Since then, no one has. So no one's measuring that damage, unfortunately. So we have no idea if there's any effect of any of these interventions on that damage. But if you're not extending maximum lifespan, presumably you're not having much of an effect on damage accumulation. I wanted to add too that the recent Ocampo paper, right, where they get death of liver cells. Why do you think they die in intestinal cells?
Oh, because they lose function. They get reprogrammed too far and they stop doing their work. Those cells, they're just much more permissive to reprogram.
So they're not partially reprogrammed? Because I don't think they were giving rise to like teratomas, so they weren't completely reprogrammed.
No, they weren't completely. Yeah, and this is
They're
actually
just
a misconception.
Two reprogrammed.
Sorry?
They were just two reprogrammed.
Yeah, they were reprogrammed too far to stop doing their work. Like the liver is no longer filtering blood, and they have markers of liver failure, ALT, AST, they're just off the chart. And so that's why mice die. And teratomas, while they look scary, they're not the reason mice die.
No, that's what I'm
Mice that
saying.
have
So
organ
these
function.
mice died, right?
Yeah.
So it's a failure of the liver to function.
Yeah, or the small intestine, yeah, basically.
Right.
And the next organ to fail.
Yeah.
So yeah, and that's basically, there are cell types that are more permissive to reprogramming. They just easily get like knocked off their cell identity and doing their function. And these are the cell types we should avoid. And usually these are the cell types like with high turnover. They're like dividing a lot, and so they're much more kind of flexible in their cellular identity. That if you start reprogramming, they just fall off to some other pathway. Neurons, on the other hand, are very resilient and resistant to reprogramming. You can induce reprogramming factors in neurons for like weeks, and they still remain neurons. They don't actually change their cell identity. This is one of the reasons why we're focusing on the brain and bio, because we think it's safer. So other cell types are just more dangerous to reprogramming. And so I think that eventually, for partial reprogramming to be successful, we need to find a way to target just the tissues that are safe to push and less likely to give us negative side effects, and yet they can produce systemic effects if we rejuvenate enough of the organs. And the vasculature is definitely one of the targets, and I think there have been...
What makes you say that you have systemic effects there? That's an assumption.
It is, yeah, obviously it's an expectation that basically if there are enough organs, like critical organs that will rejuvenate, we might see a systemic reduction in mortality risk, for example. Or, I mean, how do you measure aging? If you're not a fan of epigenetic, well, maybe that
If you want to
reduce maximum
beat
lifespan.
aging, maximum lifespan, right?
Yeah, but until then, we need some biomarkers of health, and you can look at like physiological parameters. Those mice, run farther, they have better like blood biomarkers, they have better grip strength. There's definitely some panels that we can assess short-term improvements.
That's true in healthier people. But are they aging slower?
Well, at least they tend to live longer, so usually there's
median lifespan.
frailty index is one of the
median lifespan, yeah. So, there's a huge difference in the human population between 90-year-olds. Some are fit, they're still running, but their life expectancy is still, super short.
Sure, but they
Right.
don't have interventions yet that we give them. Like if we actually give mice an intervention.
Yeah, but I'm saying that's where we're pushing the mice, is to be super like healthier towards that. But their life expectancy won't change.
Well, we'll see. That's an assumption too, that it won't make a difference until we actually make the experiment, try to rejuvenate key organs.
I mean, we do have a lot of mouse data, and you know,
Not with
again,
reprogramming. We haven't even tried this approach, like tissue-specific approach with reprogramming, like multiple organs.
You mentioned a paper where they extend by days, and it's really not impressive.
Well, 8 weeks, and that wasn't a tissue-specific approach. That was just OSK,
Yeah.
that's going to all of the cells and very.
So then the assumption is that doing it just in some tissues will have a greater effect than doing it throughout the body.
Because we'll be able to push it farther. Because right now we're kind of limited by the weakest link, which is the liver and the small intestine, so we can only induce reprogramming factors for 2 days. But if we avoid those organs, we can push reprogramming farther and expect to have more rejuvenation, and also we might.
But
be able
not
to have
like, same thing won't happen that happens to the liver. The cells won't lose their function.
Well, this is what we're observing now. Like if we just avoid the liver and the small intestine, we can push reprogramming for 10 days, and all of the mice survive, like 100% of the mice survive. So that's very strong circumstantial evidence that at least the critical systems are not losing their function. And this is, I guess, the direction where right now Alejandro, our campus lab is going to take this and investigate, I guess, what organs we should be avoiding and what organs we should be targeting. So. But I agree that there's a lot of research.
That's again based on the assumption that you're going to get systemic rejuvenation, and you don't. Like parabiosis does not give you like extended maximal lifespan.
I'm pretty sure they tried to see parabiosis.
Okay, maybe all these groups tried and they're keeping it a secret.
Well, it's hard to keep mice connected to their blood supplies for
Yeah.
years, so they only look like for short-term effects. I don't know. Any other?
I think this is we can kind of close out that section, right?
Okay.
For debate, unless any of you have any questions to each other.
Can we actually summarize the main
Yeah.
points of differences that we observed here?
I'm just curious, like how stochastic damage to like the matrix and other molecules explains the huge disparity in lifespans observed, even like among mammals.
It's what was selected for evolution, right? The proteins that make us up have a longer lifespan than the proteins that make up a fly. Right? There's a lot of differences between us.
But even like very closely related species, like rockfish is my favorite. There's like some rockfishes that lived for 10 years and some that live for 200. They're very closely, evolutionarily close, and yet they have such a huge lifespan disparity. Like what was selected? The quality of the protein, quality control machinery, but are we even talking protein, like intercellular protein, or are we talking the matrix and there's metalloproteases that remodel matrix that are much more efficient in longer-lived rockfishes than short-lived rockfishes?
Yeah, so I'm not, a fish biologist. I don't know what temperature they live at, or what other variables could affect the stability of how they're made. So I'm not going to try to debate that. But it's clear that it's sort of a misuse of the word programming. Of course, each species is programmed to give rise to an individual of that species, which has its own lifespan. Right? So in that sense, yeah, of course, the age is programmed by species. But that's not to say that, aging is programmed. Right? There are no, that we know of, pro-aging genes that accelerate aging. There's genes that accelerate behaviors that will cause you to die, like some species mate and die because they swim upstream like fish and just
It's actually a misconception about salmon.
Or-
It's the exhaustion that kills them. It's not. There's
yeah, but it's
land
behavior based. It's not. There's no genes accelerating the aging process or anything.
Well, there's obviously some program in some of the salmon, like some of our salmon that like breed and then die. There's got to be a program that activates that.
The behavior, yes.
So, it's
Yeah.
not like they're,
If they're exhausted, there's no
They're-
food,
they're not
and
exhausted,
they become
like-
prey, they're gonna die.
there's salmon landlocked in landlocked lakes. They don't swim upstream, but they still die after reproduction. After there's like
Yeah.
1 bout of reproduction. So
Yeah.
it's not necessarily behavior. It's just like in some species there
Well,
is
yeah, I
this-
mean, like spiders, that die after they give rise to their new spiders.
Yeah.
It's an evolutionary
Plants.
selected behavior so that the new generation can live. It's not accelerated aging. I mean, if you want to call it accelerated aging, then any type of behavior that kills you is aging.
Well, to me, it's the other way around. Like, there's obvious suicide programs, and aging is one of them. Just kind of like a slow, not necessarily suicide, but like slow getting rid of it.
Well, it would be great if we knew genes that did that, but we don't. There doesn't seem to exist any. I mean, all the genes in the genome have been
Could be the same
identified.
genes that are just responsible for development. It's like the speed of gestation, development. It seems to be one continuous process, at least according to epigenetic clocks, which I guess you're not a fan of, but it just seems
Yeah,
so universal among many animals.
they're just clocks. Turning back the hands of a clock does not turn back time. And in the data for the clocks, there's a contradiction that suggests that as well. Like after birth, right? That's where you see the greatest acceleration in age in the clock.
Yeah.
Right. It's also when you see a decline in mortality rate. Right? So what does that mean? It has nothing to do with aging.
Well, I think it's
It's just a clock that's measuring,
I think it's just that the first 10% of life history is actually spent on development. So it's the same path that at some point it continues and starts actually to be detrimental. But of course, in the beginning, like until the species reaches sexual maturity, mortality.
if it's a clock for your predisposition to age-related mortality, why is your mortality rate going down during that time of the accelerated, clock speed?
Because that part of the developmental program is beneficial. It hasn't yet started the detrimental part of the developmental program.
So, but it looks like it's the same clock, right?
Epigenetically, yeah.
Yeah. So
But it's the clock that just recapitulates development. And since aging is part of development, it's just the later part of development. We have this clock that essentially is continuous, but at some point, essentially after sexual maturity, as mortality starts to increase, and also the clock starts going linear. So, because the initial part of the development is like exponentially quicker.
yeah. I mean, okay, but it remains a clock. Right? I think the clock is accurate, but there's, again, no evidence that reversing the clock extends maximal lifespan.
All right. I think we're running out of time.
Oh, okay.
Yeah.
Can every speaker just summarize what he agrees with another speaker and disagrees as for now? Окей, я говорю, что один сказал, что в чем он согласен, в чем не согласен.
Нет, зачем ты это делаешь? Это нам нужно голосование. Нам нужно голосование. У нас осталось 17 минут. Еще второй вопрос.
Well, obviously, I guess we have opposite views on partial reprogramming, just how potentially successful it could be. So that's definitely a point of disagreement. And we knew this from the beginning, disagreement on the role of the matrix and the stochastic aspects of aging. I think, obviously this damage accumulation happens, I just don't think it's the primary driver. I think epigenetic changes that allow the damage to accumulate, that basically with age downregulate the machinery that's supposed to be taking care of the damage. That's the key driver to me. But in terms of agreement,
But there
I
are
mean,
no genes for the damage that accumulates. There's no repair mechanisms for that. There's turnover, but there's no repair mechanisms encoded in our genome for the type of damage that occurs with age. And it's stochastic in the sense that if you look at, an elastin fiber or collagen fiber, they accumulate, it's not the same damage in each molecule at the same amino acid, right? That's the stochasticity I'm referring to. So that's known. And it's also known that there's no machinery encoded in our genome, so it's not a downregulation of that machinery.
well, if we're talking about damage to the matrix, okay, but there's still MEPs that can remodel the matrix. In extreme cases like cancer, cancer does a great job of remodeling the matrix when it needs to. So it's, to me, it's
And at the end of life, the rate of cancer actually starts to dip because even cancer cells can't grow in that environment anymore.
quite sure why it dips.
And those metalloproteinases stop working on damaged proteins. That's also known. That's, so the normal turnover that can go on in tissues, that can't even function anymore with age. That's still not repair, but even the normal turnover can't occur.
It's interesting that you're so focused on like extracellular damage, and you think the cells are kind of secondary in that.
In terms of the aging process,
Yeah.
I think they play less of a role. There's, again, DNA damage that accumulates over time that, again, epigenetic reprogramming can't address. But
DNA damage, yeah, I think we actually agree here that it's not.
because you can take old cells and put them in a young tissue, and they behave like young cells.
And
It tells us
exactly, so
something, right?
to us it says that like,
So they're not the
ultimate function
limiting
is-
factor.
is the cells, right? The cells.
Yeah, but they're
do
not the
work.
limiting factor.
So if they're not the limiting factor, but they're the ones who do the work, then convincing them or fooling them into that they're actually younger might still be the strategy.
Yeah, I
to
mean,
alleviate these.
cells are essential to life, to our function. So is epigenetics, right, to normal functioning. But that's not to say that they're a driver of aging. That's very different. You could say that about anything in us. Everything we have in us is essential to our optimal function. You interfere with any of them, you're suboptimal. Right? That does not mean they're drivers of aging.
I guess from a standpoint of the interventions, we can debate what we think will produce the most benefit, whether it's remodeling the matrix, I guess, as you think, or replacing things, or turning on epigenetics or shifting epigenetics towards a more optimal pattern,
Yeah,
even in an aged matrix.
I think getting rid of all the damage is a higher likelihood of success.
So getting rid by just replacing them.
Yeah.
I guess ultimate replacement.
Tissue or greater level replacement. And it's also technically the easiest. I mean, for the brain, it's hard. It's going to be complicated. But, for the rest of the body, it's going to happen.
I guess it comes back to this key question whether there isn't this control mechanism inside the brain that has systemic.
I don't know where that comes from. Whose idea is that?
Well,
What's the
never
evidence for
between
that?
hypothesis of aging. I guess it comes from Mikhail Conley's father, Vladimir Dillman, like in basically hypothalamic pituitary axis or axes playing control roles in many different processes, starting from like embryogenesis to like daily circadian rhythms.
Yeah, it plays a lot of roles in that.
menstrual
But
cycles.
in aging, I don't know what the evidence is.
Well, like how does brain
Controls
work?
your weight, sleep,
Yes.
hunger.
Can I summarize and you agree or not? Like that basically you are more on the side of that aging is programmed, and you're more on the side that's like more stochastic process.
Yeah, that's what's.
Yeah. But the second is that like the
I
central
don't
role.
think aging is stochastic.
the
I
damage.
think the damage, the
Damage.
specific damage types to the proteins happens stochastically with
A-
time.
and you are more like that cells are more central to like the process, and you feel that cells is like maybe a secondary.
I mean, we don't have the test, but you know, again, there's DNA damage in cells that could be a driver. I think it's more the ECM where there's a lot more damage.
Any other important like
Стоп.
device?
Стоп.
Yeah. Then we'll stop.
Yeah, we'll stop on the here.
Okay. Good.
Говори судье, чтобы...
So yeah.
Question потом.
Questions?
No. Question второй
After
раунд.
the judges, yeah, well, yeah.
Okay. Yeah. All right. So judges will give you a moment to think and results. Yeah. So Yuri, pink, blue for Jean, green for Angel.
Oh, we have this. That's not radical.
Yeah. You guys ready? Yeah. CJ, ready? Yeah.
It should be
Yeah.
partial reprogramming red,
Partial reprogramming.
replacement
Replacement.
blue. It's not about us.
Right.
Yes.
Yeah. Sure.
Yeah, partial reprogramming.
Cell replacement. Okay. Are you ready? Let's do it. Okay.
Partial reprogramming.
Okay.
Высоко. Попроси высоко. Они очень...
Yeah.
Попроси выше.
Okay. All right.
Hey.
Yeah. So that's the replacement. Right. Right. Okay. So John, you're winning in the first round. Right. First round. First
Okay,
round.
how many
Right.
rounds are there?
Right. 2.5 versus 1.5 points. Okay. Okay, I'm back. All right. So now we can open up for questions. Yeah.
Yeah, so I guess like the major thing for me voting the way I did was that I wasn't convinced that partial reprogramming can reverse the damage that's already been done outside of the cells that are being reprogrammed. But other than that, I would like to just focus on the brain specifically for brain rejuvenation and ask one question for each of you on how reprogramming versus replacement would address that. So for replacement, like the main concern I have, like I think it's very convincing for tissues outside of the brain, as you mentioned, but I'm curious what your thoughts are on how you could reestablish connectivity if you wanted to do any sort of brain-targeted replacement, given that the brain's not really modular. So that's my question to you. And then again, brain focus for reprogramming, assuming that there is a combination of factors that can establish the desired epigenetic state in neurons and other brain cells that are equivalent to sort of not aging anymore. And assuming like in a mouse, like it's maybe a little bit simpler in terms of the tissue and penetration is simpler. And given that the whole thesis of partial reprogramming is that you're probabilistically getting that specifically right window where you get the desired effect, but not pushing it so far as to get teratomas, but maybe even like before teratomas, like other types of cancers, like neuroblastomas and things that are not fully reprogramming, but only like partially along the way. For more complex tissues, especially in humans, presumably like tissue penetration delivery would mean that different cells are exposed to your treatments for different amounts of time. So how do you think it's actually realistic to fine-tune the treatments enough to completely avoid the risk of side effects to get maximal benefit?
So yeah, no, that's a really good question about the replacing tissue in the brain. So obviously it has to be done progressively. In terms of reconnection, so all the evidence so far, and you know, there's dozens of labs who've reproduced this because of the excitement of cerebral organoids, which I'm not a fan of, but they transplant those, they transplant neural precursors. And you know, at first it was a surprise that how well they do in connecting with the rest of the brain. So as long as they're young, new neurons, newly born neurons, they'll project long distances to other parts of the brain. If you put them in the motor cortex, they'll project to the spinal cord. put them in the visual cortex, they project to appropriate visual targets. They also receive input. So we're the fourth lab that showed that if you put them in the visual cortex, over time, they'll acquire responsiveness to light stimulus. So, you know, in terms of the reconnections, they do really well. What's maybe more of a concern or more, you know, a worry is it still going to be you? Is it still going to be, you know, me? And there too, the evidence is very strong that if you do it right, you can actually remove tissue before you introduce new tissue, or coincidentally with introducing new tissue, where the functions as dear to us as personality or language can seamlessly move to a different part of the neocortex without you noticing, or the people who know you and love you most without them noticing. So there's a lot of plasticity in how our brains, in particular the neocortex, encodes information. It's super plastic over time. As long as you give it time, that plasticity occurs. So that was shown with, and continues to be shown like every day in the clinic with benign tumors that get removed. So there's a 7-year-old that had a tumor the size of an orange in his prefrontal cortex. Never changed his personality. And if you have a stroke there or a trauma that's sudden, you're a completely different person, unrecognizable. So it's really a matter of doing it right over time to remove tissue, and then adding tissue. So the examples I gave with the organoids and the single cells, even though they connect really well with the brain, the graft itself is like a complete mess, miswired, and it's not going to be processing information well. So that's where our efforts, our focus is in having the new tissue mature in a way that it's structurally fit for purpose in terms of its internal wiring, so that it can not just receive and respond to stimulus, but process it and send it in a way that's useful to the rest of the brain. So that's, I guess, how we would do it.
Doing it so gradually, doesn't that require a lot of surgeries?
Yeah, so I mean, it sort of comes down to math, you know, how much time it takes to silence tissue before you can safely take it out without losing anything. And then how long it takes for these young grafts to mature. So, you know, they're both in the range of like 2-3 years. And so and then so how much do you silence at once? You know, it's been shown, like if you can silence the whole language area, for example, that's a pretty big part of the cortex. But let's say you do this 6 to 10 times, you know, 20 to 25 years. That's what we're looking at.
Wait, 6 to 10 times, 20?
6 to 10 surgeries over 20 to 25 years to get a
That quickly?
young
Six,
brain.
only six surgeries?
Well, it looks like you can silence, you know, pretty big parts of the cortex. It depends on how much of a hurry you are too, like how old you are.
Yeah.
You could probably do more or less. But if you look at the brains of old people, they use larger and larger substrates for routine functions. So high-performing elderly are able to be high-performing because for their functions that they're exhibiting, they're using broader and broader areas of cortex. They're literally dying for new substrate to encode information. And so you could get a lot of benefit, you know, initially without doing the whole thing as well.
When you silence it, like, is there like a redundant section of the brain? Like, how does it transfer over?
The exact mechanisms of plasticity aren't really well understood. Yeah. So, I mean, we can get into that, but maybe that's.
Okay, yes. Reprogramming, not reversing damage. I think, I don't know if you caught my talk yesterday, but basically I think the main purpose of reprogramming and the main promise is being able to slow down the aging process and the process of accumulating damage. And this is what I think has been demonstrated from the very first paper by Kappa et al. Because if you look at the two mice, the treated mice and the control mice, you see that their many markers of aging or damage are better. They have fewer senescent cells, they have fewer DNA breaks, they have lower levels of inflammation, etc. So obviously this is circumstantial evidence that partial reprogramming is already having these positive systemic effects. In terms of obviously intracellular damage, partial reprogramming absolutely can reduce that damage. Extracellular damage, while we don't have like full evidence, mechanistic evidence of this improving, say, the ECM, we obviously have circumstantial evidence because the tissue histology is better. And we see this improvement in tissue histology from many papers, including not just progenitor animals, but wild-type animals. They looked at the spleen, the liver, and some other skin, but basically showing that at histological level, which obviously also includes extracellular matrix, the tissue looks better. So partial reprogramming, the way that it's being used right now with periodic induction of it, its goal is to slow down this exponential increase in mortality risk and potentially deposition of damage, so that alone can greatly extend our lifespan. I mean, reversal is awesome, and I think partial reprogramming also has the ability to reverse some of the damage, including extracellular damage, but we obviously need more experimental evidence of that. So I guess it was a little sad to hear that you think that I didn't make a good case that partial reprogramming can reverse damage. And even if we don't have evidence of it reversing damage, we have good evidence of it preventing damage from accumulating, which from a standpoint of kind of healthy people is already very, very important because that can slow aging substantially. And yesterday I mentioned that a 60-year-old person has a risk of mortality of like 1 in 100. So annual risk of mortality 1 in 100. So if we can actually kind of freeze or at least slow down this exponential increase in mortality risk, just mathematically that person, 60-year-old person, can expect to live like up to 100 years. But in terms of your question, just practical delivery issues in terms of gene therapies, obviously there are, as any gene therapy right now is facing some problems in terms of heterogeneity of penetration, and obviously you get much more concentration of the vector depositing, like around the injection site, if we're talking tissue injection. Systemic injection obviously has trouble kind of navigating to the right part of the body and also ends up in the liver. That's why the liver is right now a very popular target area for partial reprogramming companies. But none of these problems are insurmountable. There's obviously mechanisms of delivery, addressable delivery being developed, like you can have vectors that have preferential tropism to a particular cell type. You can have direct injection into the brain. You can have multiple injection in multiple areas of the brain that you want to target. And also to address the issue of heterogeneity, we and I'm sure others are thinking about implementing gene circuitry logic that will kind of monitor the level of expression in a given cell. And regardless of how many copies of the vector end up in the cell, you can have some sort of like cut off of gene expression so you don't overexpress the factors and you don't push the cells too far on the reprogramming trajectory. And ultimately, of course, we'll need to be using factors that are not human factors, but are cell type specific factors that are safer and just not able to push the cell too far in the direction that could cause problems. So I hope I answered your question.
Can I follow up? Of course. So just a question about what you mean by damage in the cell. So I think we agreed that the damage outside the cell is not addressed, but you say the damage in the cell is reversed. I don't know of any age-related damage that accumulates inside cells, other than like to molecules, like other than to DNA. What damage are you talking about?
Well, you mentioned gamma H2X as a marker of DNA damage, and this is what has been observed even in the first Ocampa paper. There's lower markers of DNA damage, double strand breaks. That is not. You're changing the whole epigenetic landscape, so expression of genes, and you're looking at one gene that's affected, that's a marker. It's not looking at the damage itself. Right? Well, it's an accepted marker of DNA breaks. So.
Yes, no, it's a very good marker of double-strand DNA breaks, right? But it's not.
Doesn't count.
Well, this is also mutant mice, right, that are defective in the repair plot. Oddly enough, not double-strand DNA repair, but another repair process that it's an endonuclease.
Lamin A, I'm not
minus-
sure that's very clear process. But
That's not lamin A. That paper was with the endonuclease mutation.
Which, no, Campbell paper was
ECCR1 or something.
No, the one I'm talking about, the original paper wasn't lamin A mice, but yeah, they also
Yeah,
looked at other
that's the
progeria.
progeria mice, but that's not where they looked at H2A plus formulation.
No, they did look at H2 gamma H2X in
that one too.
lamin A mice in the
That's
original
right.
2016 paper. Yeah.
Yeah.
And also, like histone marker H3K9 was better. I'm not sure if that's damage or epigenetic.
Yeah. In any case, it'd be good if they actually looked at the DNA
They
damage.
looked at markers in, I think, in just normal human cells as well. They also saw a reduction in the marker of DNA breaks.
In
Just
culture.
normal, yeah, human fibroblasts after partial
Yeah,
reprogramming.
again, that also was with H2A plus correlation, right?
Sorry?
That was also with H2A plus correlation.
Yeah.
Yeah, I mean, it would be good to look at the actual DNA damage. But other than DNA damage, which they didn't directly look at, is there any other type of damage that you're referring to that's intracellular?
Intracellular, well, if you remember that slide from yesterday, like full reprogramming, it alleviates all sorts of intracellular hormone activation, like misfolded
No, I know,
proteins.
but that, is that
Would you classify that as damage? Or?
misfolded proteins?
Yeah.
Yeah, I guess.
So, like, we see removal of misfolded proteins, improvement in mitochondrial function. I don't know if you consider that damage, like
Yeah, no,
worsening of
that's what I want to distinguish
Yeah.
is what's, just function, which obviously can change and is reversible versus the damage that isn't reversible.
Well, I guess I'm talking about damage in the broad sense of like Vadim Gladyshev.
Yeah, exactly. I think that's a problem in the field too, that, it's how we use these words. Damage can mean different things to different people, rejuvenation, different things.
Calls about deleterious, I guess, deleterious changes, which I guess if they're reversible, maybe you shouldn't call them damage, but
Yeah, maybe not.
from
Yeah.
the standpoint of, I guess, cell functioning better or worse, ultimately we care about the cell getting better, and if we see improvement in mitochondrial function, fewer misfolded proteins, fewer DNA breaks, markers at least of DNA breaks,
Yeah.
I think this is all.
Okay, I just wanted to know if I was missing something.
Has-
Damage.
Okay.
So the ECM of has the state of the ECM of proto-related mice been studied at all? Like, does the ECM
Separately, yeah, they looked at the matrix and
does it resemble an aged mouse? Does proto-related age the ECM as well? Because I guess
I'm not sure I can answer that offhand. I wanted to go to that.
because it would be interesting.
Not in that paper, they don't. I mean, there's a difference, again, between looking at ECM composition, and if it looks like an age signature, that's not damage, right? That's regulated expression, right? So if we're talking about damage, no, they haven't looked at that. And essentially,
Because it would be interesting, I don't know if they could do it in proto mice or some sort of in vitro experiment, but just to combine an aged ECM with a young cell and see which one wins. Like,
yeah, well, they've done, as Yuri mentioned as well, heterochronic transplants where they put young cells in old ECM, and behave like old cells. You do in several tissues of the body, and you do the reverse, and it's the outside of the cells that is dominant in terms of functionality. So if we ignore that, we won't get very far.
yeah, I mean, those are definitely interesting. I don't really actually quite understand how to interpret them yet, but like, those are entire organs too. And so, like, there could just be damage coming to them just because there's something in the blood.
These are single dissociated cells, right? Organs.
The one you're talking about, are you talking about like Vadim stuff or he
No,
transplanted?
Vadim stuff is different. Yeah, I
Yeah.
talked about it in the beginning. That
Okay.
there's like whole organs being transplanted
Yeah.
into old animals, and they're rapidly aged. And so my question is, like, how does that happen? Because the old organs already have their own ECM, so it's not like
Yeah, exactly.
there's external,
But there's something else in which there's just
which I think is signaling, like some sort of signaling.
I know, we have a collaboration with them for our brain grafts, and it's quite the opposite. If it's a tissue level, it seems to be insulated. Again, that's not the goal of just having one little piece there, but even that seems quite insulated
So,
from damage.
Could you elaborate? Like, what exactly are you observing? Like,
So he's looking at, bunch of different, he has like all these different epigenetic, what do you call them, landscapes for cortex, for brain, for whole animal, universal, like all these different clocks. So he's looked at all of them, but they all say pretty much the same thing, that the surrounding tissue is not affected by the young graft and the young, in old, when we transplant in old mice. And that the young graft is not affected by the surrounding tissue.
Really?
Yeah. And that's true in humans as well, right? So dopaminergic cell transplants, these are even naked cells, can go decades without showing the pathological hallmarks that surround those young new cells.
Yeah, pathologically for sure, yeah.
So-
But in terms of like the epigenetic age, which you don't necessarily think is a
Well,
biomarker.
yeah, I mean,
Question? Okay.
So, about 5 minutes ago, you both agreed that there's not a stable consensus definition of words like damage. I wonder if you propose one. Like, should we be using the word damage? Should we be using it to describe things that are reversible? Like, how would you like to see these words used?
Want to start?
I mean, I would like to see them as, these irreversible processes, not these enzymatic processes where they're controlled and reversible. That's just a process, that's not really damage. And it's, I don't think that that's what is a driver in that case. If it is reversible, cells are usually responding to their environment to adjust what they're doing. And so, yeah, I think damage should be restricted to this irreversible age accumulation of change.
Including irreversible with future interventions, right? With current intervention.
Irreversible in the sense that there's nothing, it's not really a biological process. It's a physical process. Like, these molecules, these proteins that break down. Like there's breaks, there's calcification, there's glycation, there's like all sorts of wide range of class of things that occur that there are no enzymes there that can fix them. So that's what I call damage. You could imagine that designing new genes that encode new machinery that can reverse all these different types of damage. So they're not irreversible, like, in that sense, although I don't think that's a viable approach. But, I'm just saying irreversible in the sense that there's, in current cells, there's no way of, in current tissues, there's no way of reversing that.
I'm okay with any classification that ultimately is just clear enough what we're talking about. Like I said, reversible types of damage, irreversible types of damage, and like what exactly are we talking about? And it's just, I guess it's better to just classify them. Like, are we talking about glycation? Are we talking about DNA breakthrough? Or this and that? But from the standpoint of how we call it, inflexible.
Any more questions from the judges? No questions?
Well, I mean, one of the reasons I voted against you was because I felt like there's like some contradiction between like how we value PCM versus so one of your arguments for transplantation like against replacement is that if you put a young brain into an old body, then maybe it might turn old, right? But then
No. I said actually if you put a young head on an old body, maybe it can rejuvenate the old body. But an old brain on a young body might age.
yeah, so it's still like because the environment might.
Now, I think there might be central signaling, which is, a big question, I guess, contention.
But then would it apply for like partial reprogramming? As in like if you try to rejuvenate all the cells with like partial reprogram all the cells, the ECM might make it not work anyways, right?
Well, this is the question, can we circumvent the ECM signaling? And so I guess this was one of my initial thesis or hypothesis that we can drown out the ECM signaling and convince the cells to function as if they're in the young ECM. And has shown things that there's some aspects of ECM that go beyond their signaling to the cell, like in the context of vasculature, for example.
So like in partial reprogramming, do that, that they can help ignore the signal from the ECM? Like, do we know that?
Well, at least just on the level of how it works, because it works by changing the gene expression pattern. And to me, that's how the cells operate. It's their operating system, right? Gene expression. And they integrate signaling from ECM and hormones and everything else. And then ultimately, what happens is this kind of little machine, which is a cell, it does its things with proteins. Proteins are driven by
Yeah.
expression.
But the ECM is intimately involved in cellular processes and the ability of cells to do their job. Maybe you can trick them. In the brain, the ECM, perineural nets, right, are memories, right? I don't know that you want to ignore that.
Alright. Yeah.
Question about the epigenetic nature of memory. I guess we'll leave it for the next debate.
Already running over time, so let's make second vote of questions, and we'll wrap it up. Discussion.
And then we do round 3.
that can be offline. In a different format. All right, so let's get a second vote. All right, are we ready?
Who's the judge?
Смотрите, как бы ни проголосовал этот судья, общая победа в любом случае.
Yeah, so I think the replacement theory gets more votes overall. So.
Здесь или
For
ничего.
a lot less money.
Alright, cool. Well, thank you everybody. This was an experiment.
А сколько я вас, ребята?